Welcome back to Shure Academy. I hope you all find the previous lecture was very helpful for your preparation. If anyone didn't watch the first lecture, see the description given below for the link to navigate you to the previous lectures. So before going into the second lecture, everyone please subscribe the channel for the latest videos of this crash course. In this lecture, I am dealing with the structure of chromosome in prokaryotes, eukaryotes, and to some extent viral chromosomes also. So without further ado, let us get into the lecture. First, let me make you sure that what is a chromosome, how the term chromosome was derived, and why the chromosomes are called as physical basis of heredity. Basically, chromosomes are tiny thread-like structures that are found to be present inside the nucleus of all animal and plant cells. Then coming to the term chromosome, it was derived from two Greek words, chroma and soma. Here, chroma refers to the color and soma refers to the body. Then, due to three main reasons, the chromosomes are considered as physical basis of heredity. That is, having special structural organization, individuality in performing their own functions, and capacity of self-replication. It was first discovered by Strasburger in the year 1875, and later the term was coined by Waldeyer in 1888. Students, don't be confused between the scientists who discovered and who termed the chromosome, because the same thing was asking repeatedly in the entrance exam. So please remember this. Normally chromosomes consist of the following components like proteins, deoxyribonucleic acids, RNA, and to some extent non-genetic structures like matrix and pellicule. Chromosomes are found to be present in all organisms in a specific number and in specific organization. Now let us understand the structure of prokaryotic chromosome. Here we can see it consists of a single giant double-stranded DNA molecule without definite nucleus around it. And one can notice that there are no proteins and RNA in the prokaryotic chromosome. Here I am giving you an example of prokaryotic chromosome which is present in E. coli, which is 4.6 megabase pairs long. Next category relates to the structure of viral chromosome. Usually they are present as a single molecule of DNA or RNA. Chromosomes are either linear or circular in shape in case of viral species containing DNA as genetic material. Example, T2, T3, T4 bacteriophages. And on the other hand, chromosomes are maybe linear in shape in case of viral species containing RNA as genetic material. Examples, poliomyelitis virus and influenza virus under the category of animal viruses, and most plant viruses like TMV, that is tobacco mosaic virus. Here we can see the viruses like poliomyelitis virus and influenza virus having RNA as genetic material and T4 phase having DNA as genetic material. Now let us understand the structure of eukaryotic chromosome in detail. Eukaryotic chromosomes are basically present in all plants and animals. As eukaryotes are multicellular, they contain more genetic information, and the genetic material does not exist as a single unit but present as many units, which are called as chromosomes. Chromosomes are located in the definite structure called nucleus. So here we can see it is an eukaryotic cell where we can see the chromosome is resided inside a definite nucleus. Then number of chromosomes are specific to the type of organism. So the number of chromosomes varies from species to species. Most of the studies told that mitotic metaphase chromosome is best suited for the comprehensive study of chromosome structure. Here I am showing you the typical chromosome structure with all of its components. So in the coming slides I will try to explain each component in detail. The first component is centromere. Centromere is the region where the two chromatids joins together, or the region where the spindle fibers are attached during metaphase. During cell division, centromere helps in the movement of chromatids to the poles, so it is also known as kinetochore. Here we can see the centromere where the two chromatids are joining together. Then the next component is chromatids. Chromatids are the subunits or each arm of a chromosome, which are normally get separated at mitotic anaphase and during a second meiotic anaphase. Then thereafter the chromatids develops as a new chromosome. Here we can see these long vertical strands or chromatids. These long vertical strands are referred as chromatids, and these two sister chromatids make up the whole chromosome. The next component is chromomere. The bead-like structures that are usually found on the chromosomes are referred as chromomeres. So one can observe them clearly in polytene chromosomes. Chromomere represents the unit of DNA replication, RNA synthesis, as well as RNA processing. Here we can see the bead-like structures that are present in the eukaryotic chromosome, which are normally referred as chromomeres. Then chromonema. Chromonema are thread-like coiled structures that are found in the chromosomes, which are associated with DNA duplication and acts as a gene holding portion of the chromosome. Here we can see the chromonema foldings that are present in the chromosome, and this is the radiographic image which shows the chromonema fibers which are present in the eukaryotic chromosome. Then I prepared a table so that everyone can understand the difference between heterochromatic and euchromatic region. So let us see the difference. Heterochromatic region is characterized by the presence of densely coiled regions, darkly stained and transcriptionally inactive regions. On the other hand, the euchromatic regions are characterized by the presence of loosely coiled, lightly stained and transcriptionally active regions. So in the diagram we can see the compactly coiled nucleosomes or DNA molecules in the heterochromatic region, which makes them transcriptionally inactive, whereas we can see the loosely coiled nucleosomes or DNA molecules in the euchromatic region, which makes them transcriptionally active. The next component is telomere. Telomere refers to the terminal portions on both ends of the chromosome. Telomeres of one chromosome never unite with telomeric end of another chromosome. Thus, it maintains the structural integrity and rigidity of the individual chromosome. So here we can see the terminal portions of the chromosome are referred as telomeres. These telomeric ends cannot unite with other telomeric ends of other chromosomes. Matrix and pellicule are non-genetic materials, where matrix is the region where chromonema is embedded and the pellicule acts like a sheath that encloses the matrix. Then the next component is secondary constriction. Secondary constriction refers to the constricted region usually found far away from the primary constriction or centromere. As secondary constriction is having a fixed position, they are used as markers. So here we can see the secondary constriction is located far away from the primary constriction and it has a fixed position, so they are used as markers. With this, I am concluding this lecture. In the next lecture, I will bring you the information about the classification of chromosomes based on various aspects. Please see the description below for the previous lectures, and please do like, share to your college friends and tell them to subscribe the channel for the latest videos. And check the channel on every alternative days for new lectures. Thank you.